Hi, my name is Kaitlyn Knapp, and I'm a scientist in the Cell and Gene Therapy Department at Eurofins BioPharma Product Testing. And today I'm going to be talking with you about my poster on the serotype expansion of our GMP platform replication-competent Adeno-Associated virus, or rcAAV method. So rcAAV testing is mandatory for all AAV derived vectors. Most of the testing is based on AAV2 or AAV serotype 2, because that's what's currently in the market mainly. However, new clinical studies have shown a lot of promise with other serotypes, which has created a need for testing of these serotypes as well. So to look into how we could possibly expand our current methods to meet this need, we first wanted to identify variants between the serotypes that could lead to potential testing problems. So we performed an infectivity study using AAV5, 8, and 9. And this was done with HEC293s, HG1080, and HeLa cells at various concentrations using GFP-expressing virus so that we could monitor infectivity. And we found from this a great variance between the serotypes in cell line susceptibility, infection sensitivity, as well as response time. So for cell line susceptibility, we did see that all the serotypes were able to infect across all of the cell lines. However, all the serotypes greatly favored the HEC293 cells. This was really excellent news because our current method utilizes HEC293 cells for its cell-based portion. We also saw a great variance with our infection sensitivity. AAV5 specifically had a lot less infection success than the AAV8 or AAV9. And you can see in the infection study section of my poster images of AAV5 and AAV9 on the same cell line at the same concentration. So even though there's positivity in both, there's definitely a variance in infection. And we also saw that AAV5 had a longer response time than 8 or 9, while AAV9 showed infection success within a day post-inoculation. AAV5 showed no success at those lower concentrations, and even when tested at higher concentrations, took multiple days to have full infection success. So using the variants we found for the study, we wanted to implement these serotypes into our current rcAAV method to see how it would work with them as it stands. So we inoculated AAV5, AAV8, and AAV9 onto HEC293 cells with Ad5 helper virus as per our current method. They then underwent multiple subpassages and then were harvested for analysis with qPCR. And this is done with a target of rep2. It should be noted that all of the viruses we used in these studies were modified to have rep2 so that we could analyze them. And we found from this that all of our AAV9 replicates had rep2 positivity in them. However, we did not see any rep2 positivity in AAV5 or AAV8. It is likely that this is because of the variance in infection sensitivity and response time that we saw in the infectivity study. The response time being longer for these viruses might mean that they did not have time to properly amplify during the subpass periods. It also could show that the concentrations are too low for testing these viruses based on, especially AAV5 was never shown any positivity at this low of a concentration. But because of our success with AAV9, we then moved on to looking at how to optimize our AAV9 testing and using AAV9 as a positive control in this. So we tested AAV9 in our rcAAV assay at 3 concentrations and had 2 out of 3 of the concentrations show positivity, which is really amazing. So based on this success, we're now looking to optimize these rcAAV9 methods and then looking further into bringing in the expansion of rcAAV5 and rcAAV8 methods based on what we saw in the infectivity study, and then even moving on to other serotypes so that Eurofins BPT can meet the client and industry needs.